Dear all, hello. I like to take first the opportunity to thank the organizers and in particular Professor Da Silva to allow me to give a talk at this nice conference. I talk about flow chemistry and plasma for nanomedicines and health nutrient extraction, and this will be evidenced by tests in and for space. I'm at the School of Chemical Engineering at Adelaide University in Australia. I'm a program chair of the ARC Centre of Excellence, Plant for Space, and finally, I'm the research director at the Andy Thomas Centre for Space Resources. I do since 32 years microreactor research. This is today known as flow chemistry, but I'm also deeply involved now in space research, as evidenced by the title of this talk. I made the first Australian-led NASA ISS experiment in 2022, but I'm still very busy with flow chemistry as evidenced by this title cover. The motivation of this presentation is, why do we actually need space medicines? We might not need space medicines when we go for short-term travel at not too long distance as to the International Space Station, as you see on the left side. The medicines are supplied from Earth. But when we go further and further into the deep space, when we go to Mars finally or have a moon colony, we might have to make medicines in space. There are medicine vulnerabilities. Earth-designed medicines can fail in space because of degradation, of microgravity effects, temperature swings, not to forget that diseases are different in space and therefore demand their own medicines. So space tailoring of medicines is essential. This research is motivated and supported by the ARC Centre of Excellence, Plant for Space. So let me introduce this. On the right side, you see a graphical vision of how we might grow plants in space in vertical farms. Plants for Space has 4 missions. The first one is complete nutrition of plant-based foods. The second one is about zero waste, optimized for controlled environments. We want to make the sustainability better in space than we did previously on Earth. And finally, we want to reprogram plants to be biochemical reactors to serve for on-demand bioresource production. But not to forget, we have a strong human factor and we want to help the next generation of space researchers. So our fourth mission is future-ready workforce and society. We like to be in contact, and this is one of our deliverables, with many hundred PhD students, up to 1,000 PhD students, and we are on a good way to that. I think you all know that fluids behave different in microgravity, and that makes it so exciting, especially for our community interested in flow chemistry. Particles do not settle, suspensions therefore have a different behavior, gas bubbles do not rise, and droplets also show a different behavior. The space medicines under consideration in this presentation are nanoemulsions. They are made out of oil, water with buffer, and a surfactant. Their individual concentration determine how the nanoemulsions looks like. Dependent on the point in the ternary phase diagram on the left side, we can have anything from isotropic liquids to transparent gels to stable emulsions and to biphasic systems. This slide shows how we make our nanoemulsions. First of all, we have to be sure that the drug under consideration is sufficiently soluble. We consider melatonin as a drug, as this is against osteoporosis, which is bone loss experienced significantly by astronauts when they are in space. Melatonin solubility is determined in oil as shown in the figure, and various oils are considered. You see that the solubility in canola oil, for example, is not so high. We consider the MCT oil that gives the best solubility. And the MCT oil has also the desired manageable viscosity. It's also an important point when you go for a flow system naturally. Now we need to look for the hydrophilic lipophilic balance, which is so important when designing emulsions. We go from the picture to the upper left to the lower right. So first we prepare surfactant blends, and then we look for the right hydrophilic lipophilic balance HLP. And if that is not sufficient, as you see in number 4, we adjust it by choosing different surfactants. One is span, and the other one is a twin surfactant. After this, we mix the system with an impeller, and we make a stability testing if we get a stable emulsion which shows no creaming effect. And then we determine the hydrophilic lipophilic balance. And that the choice of surfactants is crucial to get the right HLB. We use conveniently a mixture of two surfactants which were mentioned already, twin and span. And we have different varieties of those surfactants which are called twin 20 and twin 80, and we use only span 80. Now, when mixing twin 20 and span 80, we get a behavior as shown in the left figure. Plotted is a creaming index versus the composition of these surfactants which determine HLB. At an HLB of 11 and 12, we have zero creaming. Left and right, we observe creaming. So we don't have a wide window, it's rather narrow. When we use twin 80 and span 80 mixture instead, we see that above an HLB value of 11, no creaming is observed, so we have a wide operational window. Let us have a look at the physicochemical properties of the nanoemulsions we created. First, I have to inform you a bit about nomenclature. We have blank nanoemulsions which are uncoated. Then we have chitosan-coated nanoemulsions, and we have the same thing uncoated and coated for nanoemulsions which contain the drug melatonin. On the left side, there are 5 subfigures given, and they have 6 bars. We have 4 different emulsion. So the 2 other bars come from the fact that we give chitosan in different concentration. So virtually as a thin and as a thick layers. Now let's have a look. We start with A. A shows us the droplet size, and we can see that it increases a bit when we add chitosan without the drug. Interestingly and surprisingly, it decreases when we add the drug. This effect is not well understood as now. Now, if we look at the polydispersity index, then we see that the polydispersity index increases a bit when we add chitosan. This is understandable as we have a well-defined system. We add something which is in itself also well defined, but the addition has then still a larger variability, so we go a bit down with the polydispersity index. The theta potential is an interesting one. It determines stability, but stability only driven by electrostatic forces. Now what we see is when we add chitosan, with melatonin and without, the electrostatic stability goes down. And that was expected because we neutralize, chitosan is known to neutralize charges. But chitosan adds a steric factor. It's a rather rigid coating, and this is not reflected in this experiment. So we are still optimistic to have stable chitosan-coated emulsions. Finally, viscosity is an important point. When we add chitosan, we increase viscosity, that is clear. And when we add a lot of chitosan, we increase viscosity even more. So there's certainly a limit when you cannot use this nanoemulsions anymore in flow. The pH effect I do not discuss. Is very important for Earth and space medicines. We expose the nanoemulsions to thermal stress, meaning we rise temperature. And you see that both coated and uncoated nanoemulsions only decompose at really higher temperature above 40 degrees. Now, under simulated microgravity, and we have a microgravity machine, you can see that within a number of days, the droplet size does not change for the coated nanoemulsions, while we see a slight change for the uncoated nanoemulsions. That is why we coat it, and we can prove the effect is given. We were finally so lucky to have a sounding rocket experiment with the German space agency DLR in the frame of their MAFOIS 15 mission. Our experiment has been termed StarMate. You can see that there's a whole big supply chain behind one rocket experiment. There are weeks and months actually of preparation in advance and weeks afterwards until finally you can publish the results. I do not go into details here, but it shows a bit about this procedure, which is highly standardized. Here are the results of our sounding rocket experiment given. First, I like to show you a cross-section of the rocket, which you see on the left side. What you majorly see, all the white parts, that as equipment which we didn't use. This is from another user. Luckily, there's a recess, and in this open void, we place our sample. It's a black cartridge with 4 bright spots. It's on the top part of the cross section. On the right, you see the cartridges from the side and also from a top view. And from the top view, you can see the 4 samples which were included. Now, if we look on the results, what did the flight actually do with the emulsions? We also have a shaker test, which is standard in such space investigations. But let's look at ground sample and flight sample. We see a difference in the non-coated emulsions. The coating stabilizes it. So we can conclude that coating is needed to exclude any microgravity or other flight effects. If we look at the polydispersity index, we see the same. We have a low one, which is roughly the same on both sides, but in the case of the non-coated emulsions, the polydispersity index increases. Yeah, look at the 3 bars in the middle. The theta potential, which again is electrostatic contribution to emulsion stability, increases interestingly in the flight. That is maybe our most significant finding. The emulsions seem to be more stable under microgravity conditions. Naturally, we need to prove that it's really true to microgravity and nothing else. Is a drug release profile, so the release of melatonin out of the nanoemulsion droplets, and we see it's quite fast when we have no coating, and when we apply coatings, then it's lowered down. This is in line with the idea of controlled drug release, the reason for which we form those coated nanoemulsions. I do not want to comment for reasons of time on further details. We come to the second part of my talk, which is about getting health nutrient out of plant grown in space and for space. Take a tomato bush as shown over here. We have edible food and we have non-edible, which is waste. Now the non-edible parts, this is the one which we extract because we want to have a circular process which creates no waste. The solid part of the waste, we favorably extract by batch. Solid liquid extraction is not impossible in flow, but it's maybe not the first choice. We use flow where it's strong by liquid-liquid solvent extraction to come from a mixture of health nutrients to hopefully one health nutrient at a high purity. We use plasma to open the cell walls of the waste. I show this in detail on the next slide, and we use plasma also to prime seeds, which I will not show in this presentation. Now, before showing the plasma effect, I like to give the detailed process scheme. We start with 1,000 kilograms fresh tomato. We go to 50 kilograms pomace, and we use part of it for food and part of it for the health extraction. We consider a fast extraction in batch, as I said, by supercritical CO2. Why? Because for this, literature data are available for processes with lycopene, carotene, and tocopherol. For the other four health nutrients shown here, we do second extraction, aqueous ethanol, which gives the molecules shown over there. These are hydrophilic health nutrients like quercetin, for example. You see, you get different amounts of the health nutrients, so this can vary virtually from 0.25 grams up to almost 50 grams. Now we have chosen these molecules deliberately. Why? Because they present molecules of high economic value and also of a high demand by people who have a sickness. Now, the special thing about this extraction is that everything which we want is closed in a shell, and the shell is the cell, so we have to break the cell walls. We know existing extraction processes, but they have set disadvantages, especially in breaking the cell walls. So for this reason, we investigate non-thermal plasma and also ultraviolet to break the cell walls. Now, the mechanism of breaking the cell walls is in parts known, and it has to do with locks which connect cells to each other, and those locks are glycosylic bonds, and in particular the 1,4 bond connection, which we have to break by plasma. And there's large literature evidence on this, that this is working well. There's also a little bit literature evidence on UV hydrogen peroxide breaking of the cell walls, but I do not go in detail on this. Now let's look on the plasma. This is our plasma reactor, a plasma jet reactor, and we use argon gas here. This is a laboratory investigation, and in industrial process we would not do that. Now, if we look at SEM pictures, we see there is large changes, very evident for spirulina, but if you look closely on chlorella as a microalgae, as a cyanobacteria, sorry, not microalgae, you see that it's quite changed after the plasma treatment. And we can also see this in the FTIR spectra. You have to look a bit closely in it. The black line and the red line, they don't coincident, especially with the amide font and the polysaccharides, which I highlighted before. Let us have a look at the plasma effect at the example of beta-carotene extraction. The plot shows left a control experiment without plasma pretreatment, so only extraction. And on the right side, we see plasma pretreatment followed by solid liquid extraction. It is visible that much more beta-carotene is extracted when we open the cell walls by plasma, and that is also given as a plot here. And the table shows nicely the magic of flow. If you use it as a black box, then you might end with the scenario number 2, so a space-time yield of only 0.5. But if you use this as a flow chemist will do, at the best operational point, you can have 7,000 space yield, time yield. So that is essentially the difference of a good flow process 7.5 versus 7,000. Quercetin is a polyphenol and it is an anti-inflammatory agent. So we also extracted quercetin in batch after opening the cell walls with plasma. And you see on the left side that for spirulina, we get 25% increased quercetin release. You have to compare the blue curve, the dark blue curve, with the red curve. And for chlorella, this is a 22% increase. As chemical engineers, we like to compare our results to models. A common one is a Pallox model. Without going in details, you see that for the low reaction extraction times below 3 hours, there's a big difference between the model prediction and the experimental value. And this is because diffusion is not explicitly considered in the Pallox model. So we also looked for the power law model, knowing this has a diffusional component. And now the results fit much better. Where does the diffusion come from? It is because we have to enter inside and outside of the cells, and within the cells there's only diffusion of the extraction component. This is very much different from a normal extraction. Now, we also like to see the function of the quercetin, not only to extract it, but to make sure it does what it is supposed to do. And it is an antioxidant. So we use the standard technology with both spirulina and chlorella, and we could show that with plasma, we also get more antioxidant power. We also used another plasma equipment of my colleague Brian Cott. It is larger, so you can scale up and you can operate under reduced atmosphere. That are the advantages. And we did it for beta carotene extraction. And you can see that we also got here a major shift from 267 milligrams per liter extracted to 372. Now I come to the flow part. We were successful in removal of non-polar impurities. Quercetin is here on the left side because it's a polar molecule. And you see there are quite some major impurities, more for chlorella than for spirulina. And by liquid-liquid solvent extraction, so by segmented flow in our coiled flow inverter, we could show that the plasma-treated samples are more pure, so have more impurities removal than the non-plasma-treated samples. Finally, we also had to check with the flow experiments that we didn't lose quercetin. And here you see we have high recovery rate for the quercetin, both for the untreated and for the treated, plasma treated. Thank you for listening to my talk. I like to end my talk by pinpointing at certain publications which we did, which are relevant for this talk. The first one is busy with microfluidic engineering of chitosan-coated nanoemulsions for controlled release and stability in space pharmaceutics. I also made a paper which describes the effect of simulated microgravity of a random positioning machine. Is that true microgravity, or are there some effects which may falsify the results? The base for the work shown here was laid in the next paper, which is on microfluidic spontaneous emulsification for generation of oil-water nanoemulsions. To show that our research is also good for real-world application, we demonstrated this in the use of the emulsification for sunscreen applications, a publication done together with Unilever company. Also, to give the broader context, we made a review on nanofood process technology. And again, for a broader context, we made a review on microfluidics and macrofluidics in space. Finally, a very recent paper describes what I've shown in the first part of my talk. It is about pre-treatment of plant biomass waste, and the pre-treatment methodologies are non-thermal plasma and microwave. Thank you again. It was very nice to give this talk.